speaker, Siavash Mirarab, and he is also a longtime CGSI participant, a faculty fan. Definitely really memorable having him around CGSI, and really happy to have him back, giving us a talk. Again, he was actually gave one of the best, give many good talks, but one of his talks was highlighted as one of the best talks. Really looking forward to hearing what he's going to say.
Thanks, Aliza, for that introduction. I also missed the opportunity to be in CGS this year, in person, but this is also very nice to be able to see everyone here. So I'm going to talk today about the problem of phylogenetic placement. Everyone here is at some level familiar with what I'm going to call de novo phylogenetic reconstruction, which is you have a bunch of sequences, you infer a tree from it. That's the traditional, that's what you're familiar with. Phylogenetic placement is a similar problem. You have your sequences, but you have two sets of sequences. You have backbone sequences, or we call them sometimes reference sequences, and you have query sequences. On the backbone sequences, you already have a tree. You're happy with that tree. You have to spend a lot of computational time inferring it. You think it's accurate. You don't want to change that tree. All you want to do is add the query sequences on top of the tree that you already have, right? So you just want to place the queries on the existing tree. Now, why would you want to do this? There are benefits and shortcomings to phylogenetic placement. The benefit, the obvious one is running time, computational, so you're not re-inferring the relationships you have already inferred. But also, we usually place these queries independently, one at a time, independently from one another. So that makes the whole placement process, increase linearly. The running time would increase linearly with the number of queries you have. It's going to be embarrassingly parallel, so it's going to be much faster. What's probably not so obvious is that it can even improve accuracy if the queries that you have error or missing data. If you include them in a de novo reconstruction, those errors or missing data in the queries can impact the relationship between the existing sequences that you have, the references, and maybe negatively impact them, right? But in the placement, they don't impact other queries or the references, so the overall accuracy might go up. And the flip side of that same coin is that you will not be able to improve the relationship between your backbone sequences now that you have seen more data. So that's a shortcoming. And the other one is, if you're placing these queries independently, you won't know the relationship between them, right? So if the correct placement of another query is here at the sister to the query one, you would get it as a sort of independent branch on the same place. So some benefits, some shortcomings. People have thought about phylogenetic placement since 80s, but not sort of as its own, as part of a greedy algorithm that would build a phylogeny starting from a small one and just like adding one new sequences to it. Since around 2010, there has been much more attention paid to phylogenetic placement. Two methods came out around then, EPA and by Alexis Stamaitis, and P-Placer by Eric Matson. And these both do maximum likelihood placement. And the reason all of a sudden phylogenetic placement was so much more important was because of its new applications. One of those applications was microbiome analysis. So we had started getting a lot of this microbiome data where you have a set of unknown sequences from unknown organisms. You don't know what they are. You have these mixtures of unknown 16S or metagenomic reads. You want to know what they are. One thing you can do is add them to a phylogeny of known species, and that will help you identify the species, and it will also help you do a lot of interesting downstream analysis of these sequences that takes into account the phylogenetic relationships. Not so different from like what Sohini was talking about. Instead of like in terms of ancestry, here is about sort of the larger scale evolution. You can take that into account in your downstream analysis. If you're interested about what you would do downstream, this paper by Eric Matson from quite a long time ago, 2015, is a Very nice paper to look at. Okay, so that's one application, and more recently, phylogenetic placement has been used a lot in molecular epi. So you want to know who transferred the virus to whom, and you can place your sequences. So you might be wondering who infected the president of your country. You can get their sequences and place it on the tree, and that will tell you something about the transmission. And so that's another application. Now, what you might be wondering is, why do we even need phylogenetic placement for these applications? Can we just use BLAST and find the closest species in the reference set to a query that I have? And of course you can do that, but there is that kind of methodology starts to be much less accurate when you have differences in the rates of evolution across different lineages. So if you think about this tree, this phylogenetic tree, and so branch lengths here are sort of proportional to the amount of change in each branch. The closest species to this query is this species B, right? But that is not the closest species evolutionary. Evolutionary C is closer. So if you just go with the closest match, you're going to be misled here. Like you might infer the wrong transmission event here in the case of molecular epi. We looked at this in simulation. So we had the simulated data set lying around of 200,000 RNA sequences that were simulated using a complex model. So we just removed sequences from it and we tried to insert it back. So it's a leave out kind of experiment. And we asked what percentage of time, if we just choose the closest sequence, what, for example, BLAST would do, what percentage of times do you find the correct placement? And we saw that between 50 to, I don't know, 60% of the time, depending on, so as you go from over here, if you use the full reference set, and then we subsample the reference set randomly to get sort of less dense references. And if you have the somewhere between 50 to 60% accuracy, depending on how dense your sample, your reference set is. Now, is this the best that can be done? And the answer is no. If you use those phylogenetic placement methods that I mentioned earlier, EPA and P-placer, maximum likelihood, you can get much higher accuracy. And so that's great. And that's because they know how to deal with situations like this. Now, the issue here is that these methods are, of course, a lot more difficult than just finding the closest match. So depending on which method you use, we couldn't really scale them beyond references with 10,000 species. Memory becomes a problem, running time becomes a problem. So that's sort of the limit of their scalability. But we see very clearly that more dense reference sets give you better accuracy. This is not a surprise to anyone. Everyone knows that the denser your reference, the better, if you can use it. So are these super large reference trees that we are talking about here fantasy or real? They are very much real at this point. So we have had trees with hundreds of thousands of 16S sequences in them for a long time. And more recently, since like the last 2 years or so, we have started getting many genome-wide trees with tens of thousands of species. These came out in the past like 2 years. In the next couple of years, I predict we're going to have a lot of trees with hundreds of thousands of bacterial genomes in there, right? So these large references exist. Now the question is, how do we actually use them for placement? There are several groups who are working on phylogenetic placement on very large trees. This is the approach that we took. We said, why don't we go back to distance-based phylogenetics, which is more scalable than maximum likelihood? And the idea is very simple. You have your query, you have a bunch of sequences, you compute the distance between the query and all the sequences. You can phylogenetically correct these distances using models like JuxCanter. So these are now phylogenetically corrected distances. If I come up with any potential placement of my query on the backbone tree, I draw a small tree here. It could be much larger, but for whatever tree, if I come up with a placement, that placement implies also a distance vector. You can compute the distance between your query and every reference on the tree. So the path length, that gives you a distance vector. And you can now define an optimization problem to minimize the sort of the distance between these two vectors, right? So the L2 norm, for example, right? So so now you have an optimization problem. You basically find the placement that maximally matches with the distance sequence distance. A naive solution to this optimization problem would require quadratic time. You have to look at every branch and compute the best placement. But we were able to come up with a solution that increases with like a linear time algorithm, linear with respect to the size of the reference tree. And this algorithm was called Apples. It's just like dynamic programming. You have to do some bookkeeping, and then you can do it in linear time. Just a couple of weeks ago, we put on archive an updated version of it called Apples 2. It's the same idea, except it doesn't compute all distances. It uses divide and conquer on top of the dynamic programming to have sublinear running. Okay, so that's the method. Now, how well does it work? This is the same simulation I showed before. Apples 2 is added to the picture. And you can see its accuracy for the data sets where you can run the maximum likelihood methods. It's as good as one of the maximum likelihood methods. It's a little bit less accurate than the other one, P-Placer, on the sizes where they both run. But you can, of course, run it all the way to 200,000 sequences in your backbone. And as you would expect, the accuracy keeps going up. This is simply just the benefit of better taxon sampling in your reference. In terms of running time and memory usage, Apple, distance-based method uses a lot less memory and a lot less time than the maximum likelihood methods. Okay, so Apple 2 can sort of enable us to use this very large backbone trees. Now, this is just like a simulation with one gene. Can we, let's see how it works, or does it work for analyzing actual microbiome data? So to answer that question, again, we need some simulations so that we can measure accuracy. So what we did was we took one of those reference trees that I talked about. We chose this one because this is the one we had inferred, so we were familiar with it. This tree has 10,000 species. It was inferred from 380 marker genes. So from this tree, we chose 200 species at random. We removed them from the tree, and we used these as queries. So we simulated reads from them. Then to analyze them, to place them, we basically assembled the reads, right? So now we get the scaffolds. These scaffolds are not going to have the entire genome. They're small sort of portions of the genome. They're going to have very varying number of genes. We looked for these marker genes that we have inside this scaffold. We found varying numbers of them. However many we found, we concatenated them. And okay, and then we tried to we placed that concatenated set of genes on this species tree using Apples 2 method that I mentioned. What you see here on this figure right here is how far away from the correct placement we are in terms of number of edges. So 20 means that you are far away 20 edges from the correct placement. 20 edges away from the correct placement is very high error, right? Because remember, like, with a tree that has 10,000 species, its entire diameter is, in the order of log 2 of 10,000, right? So it's not much better than random placement. It's a little bit better than random placement. So this is very high error, but the x-axis here is showing how many marker genes one of these scaffolds included. And what you see is that as you get to somewhere around 15 marker genes, the accuracy starts to get much better. The error goes down. By the time you get to something around 40 marker genes in the scaffold, we have very good accuracy. So large scaffolds that have like 40 marker genes or something like that, they can be placed with very good accuracy, not these smaller scaffolds with few genes. And there is a lot of variation around. So the blue bars show 95 percentile of the errors across different cases. You have a lot of variation. Like these small, even with 1 or 2 genes, sometimes you get the right placement, sometimes you get very wrong placement. So it really depends on the gene. Why does it depend so much on the gene? Why is the accuracy so low here? I mean, the gene still is a lot of information. The answer has to do with what I've talked about in CGSI many times before, the issue of gene tree discordance. When you have only 1 gene, so remember, we are placing on the species tree here, not the gene tree. And as a lot of you know, the gene trees and the species tree can be different. Across the genome, the evolutionary relationship between the species can change. And so if this is your evolutionary relationship, so the data are generated on the gene tree, but you are placing on the species tree, none of the algorithms that we have are designed for that. So they're going to give you the wrong placement very often. Now, if you have a lot of genes, then maybe they start to sort of cancel each other out. And that's why with many marker genes, things look much better. Now, this led us to a follow-up question. What if we have, in the extreme, what if we have only a single gene? Can we place a species on the species tree with a single gene? For example, you have 16S data. Can you place the 16S data on the species tree? You can't do that with perfect accuracy. There is not, you can prove that that cannot ever be done with perfect accuracy, but can we do it with reasonable accuracy? And if we can, then there is there is an interesting benefit, which is you can take 16S data and metagenomic data, the 2 modes of microbiome data that we have always had, and they have kind of always been more or less separate, and put them on the same tree and perform downstream analysis. So to solve this problem, we recently came up with the machine learning kind of approach. So the idea is this: you give me a tree, this would be a species tree, you give me sequences from 1 gene, we train some model, I'll tell you how, but the output of this model will be a sort of, this model is basically a function that given a sequence will embed it in Euclidean space. So it's just like an embedding of sequences in the Euclidean space that we learn. Once you learn that embedding in Euclidean space, phylogenetic placement becomes trivial with apples, right? You can just compute, let's say, the L2 norm between these, the L2 distance between these vectors. You can use those as distances. And once you have distances, you can place things on the tree as I described earlier. So the placement becomes easy. The question is, how do you train this, this embedding? And the answer is that a phylogenetic tree defines for you distances between your sequences. So I already have a distance matrix based on this phylogenetic tree. Any embedding will also define for you a distance matrix. My loss function, when I'm training this model, what I'm trying to minimize is the difference between these 2 embeddings. So that's sort of the loss function here. How closely my embedding matches the tree that I'm given, in this case, the species tree. Now, what models you put here, I don't think there is any like theory to figure out what's the best model. We just tested a bunch of things, including, and we found a CNN model, a convolutional neural network worked pretty well. I'm not going to claim that's the best way to solve it. There might be better models out there. When we tested this on that tree of life data set that I showed, so I said we have 381 genes. We chose 50 of them. We chose some of them that are very different from the species tree, some that are very similar to the species tree. And we asked ourselves, given data from only that gene, how accurately can we add a query to the species tree? If we use the traditional models like Juice-Cantor, this is for computing distances. This is how much error we have in terms of number of branches. This machine learning approach I just described, they call the method depth. That is going to give you much better levels of error. So there are 2 or 3 genes here that remain difficult. But for the remaining genes, or errors is close to 2 or 3 edges, and that's like tolerable levels of error. You're 2 or 3 edges away in a tree of 10,000 leaves, so that's not bad at all. These are of course on testing data that are not in your training set. If you look at like the same species that are in your reference set, you have close to perfect accuracy. That's not surprising. You could also, this is the last result I want to show. So if you take this 16S gene, for example, oftentimes we don't have the full length. On the full length, we have something around 2 edges of error on average. Oftentimes we don't have full length. If you only have 2 regions, V3 and V4, that they combined, they're around 400 base pair, you can get levels of accuracy that are almost as good as the full 16S gene, right? So, and this you can get with amplicon sequences. You can get V3 and V4 combined with amplicon sequencing, and so you can update the species tree with amplicon sequences with what I would call reasonable accuracy. If you have only 1 region, 100 base pairs or 150 base pair, the accuracy goes down. Your placement error is more like 4 or 5 edges. That to me is a little bit high. So, but with V3 plus V4, I think is reasonable. Okay, so that's everything I wanted to show. I have in my slide a list of various papers that I mentioned. I'm going to make these slides available on my website right after this talk if you were interested. And with that, I want to just mention that a lot of this is work I did with my students. Some of it is with my former advisor, Tandy Warner, and a lot of it was in collaboration with Rob Knight's lab here at UCSD. Thank you. Okay, so let me stop sharing. We go to questions, Eliezer.
Yeah, sorry, my button wasn't working on the unmute. So yeah, let's go ahead with a few questions. Great talk.
I have a question, Siavash. Very nice talk. With the phylogenetic placement problem, is there any approach that tries to do the gene tree, species tree reconciliation, but just with placement?
Yes, we published a method called install. Basically, you have to first update the gene trees using one of these machine maximum likelihood methods or whatever method, and then update the species tree by looking at the set of updated gene trees. And for that second step, we have published a method called install. It does that kind of reconciliation that you are talking about. Those are a little bit harder to use in practice because you have to first update all the gene trees and then the species trees. Just the pipeline is a little bit longer.
Mm-hmm. And but do you see any benefits compared to like the simpler distance-based?
Yes, they definitely. So in this paper on DEV, we also have results on that approach. That if you have enough genes, if you have more than, I think, 16 genes or something in the analysis that we did, you definitely have higher accuracy with that pipeline. So those results are in the archive paper. You could look them.
Okay, great, thanks.
Okay.
I have a question, Siavash.
Go ahead.
Have you explored what happens if you have admixed samples?
So in the case of microbiome data, admixture not per se, but of course we have horizontal gene transfer, right? So all these species that we have used as query, they differ from the species tree by horizontal transfer mostly. And that's, I think, one form of, I guess, admixture. So in that sense, yes. But if you're talking about like actual admixtures of two populations, no, we have never tested that specifically.
Okay, great. Well, thank you so much. Thank you so much for that talk.